Good afternoon to all. I'm Shalonida from Emmanuel Ersel College of Pharmacy. Today we are going to discuss about identification of bacteria by staining techniques. Introduction. Bacteria are microscopic organisms. They are also colorless for most part. In order to visualize them, to study their structure, shape, and other structural characteristics, it is necessary to make them more visible. First, we have to know what is stain. Stains are dyes or agents used for differential coloring of microorganisms to observe their structure under the microscope. It is an organic compound consisting of a benzene ring. Stains are of three types: acidic dyes, basic dyes, and neutral stains. Acidic dyes are basic dyes. First one, acidic, anionic compounds, and they are more alkaline in nature. Example, picric acid and eosin. In basic dyes, they are cationic compounds and acidic in nature. Example, methylene blue, crystal violet, and safranin. In neutral stains, they are the combination of both aqueous solution of acidic and basic dyes. Staining techniques. It is categorized into two types: simple staining and differential staining. In simple staining, the dye methylene blue or crystal violet is used for both positive and negative stain. In case of positive staining, methylene blue is a basic cation which have positive charge attached to the surface of the object, and it is a negative charge. In case of negative staining, it is a basic anion having a negative charge attached to the surface of object, and it is positively charged. Example, picric acid and eosin. Differential staining. In this staining method, we are using more than one reagent or dye. It is added to specific object. Example, gram staining techniques. Gram staining technique involves staining of gram-positive and gram-negative type of bacteria. Gram staining of bacteria. It is discovered by a Danish physician, Dr. Hans Christian Gram, in the year 1884. He differentiated the staining procedure for two distinct groups as gram-positive and gram-negative bacteria. In this technique, there are 6 basic steps involved: smear preparation, heat fixation, staining by crystal violet, Lugol solution, iodine with acetate alcohol solution, and saffron. Procedure. Staining is related to physical and chemical difference in cell wall of bacteria. In gram-negative bacteria, the cell wall is thin, multi-layered with lipids, and they are dissolved in alcohol, which results in formation of pores in the cell wall. So there is leakage of cellular contents, and it is happened by crystal violet iodine complex. Discoloration of gram-negative bacteria possess pink or red color due to saffron. In case of gram-positive bacteria, cell wall is thick and composed of protein with mucopolypeptide. On application of decolorizing agent, dehydration takes place due to crystal violet or Lugol solution. This results in closure of pores in the cell wall, so there is no leakage of any cellular content. Gram-positive bacteria possess purple or violet color. Acid for staining. This technique was developed by Paul Ehrlich and modified by Seale-Nielsen. It is also known as Seale-Nielsen staining technique. It is used to identify the members of Mycobacterium, especially Mycobacterium tuberculosis and Mycobacterium leprae. These organisms are difficult to stain by means of normal staining method. So, to overcome this, acid fast staining is used. Can be classified into two types as acid fast and non-acid fast. Acid fast retains their stain and non-acid fast do not retain their stain. The reagents used are carbonyl fusion reagent, acid alcohol solution, sulfuric acid solution, and methylene blue. Procedure. First, we have to prepare a smear and heat fix on a glass slide and place carbonyl fusion reagent. Heat it gently for 5 minutes and allow to cool, and then wash the slide on tap water and slightly add acid alcohol solution and allow it for some time. Then add 2 to 3 drops of methylene blue and let it for 2 minutes. Again wash it in tap water and focus on oil emulsion objective. The organism will appear bright red on a blue background. Thank you.